This is a continuation of the previous video where we have been dealing with how proteins are translocated to mitochondria. In the earlier video, we looked elaborately at the structure of mitochondria. We looked at the various functions of mitochondria, and we have also looked at how mitochondria, the genesis of mitochondria. Now there is one person whose contributions we must recognize. That is a scientist from Switzerland. His name is Godfried Schatz. Gottfried. So, he is a man of mitochondria. He made certain wonderful, breathtaking discoveries in the 1960s. In 1964, he discovers that mitochondria has its own genetic system. Subsequently, he discovers that mitochondria are originated from a pre-existing mitochondria. Then he also comes up with the hypothesis that proteins are translocated to mitochondria based on a particular mitochondrial signal. So these are some of the contributions done by Gottfried Schatz, and we are going to look at in this video how exactly proteins are translocated to mitochondria. We will give a lot of importance to a number of crucial experiments which led to what we know today. We are looking at the possibility of a signal that will direct a mitochondrial protein which is synthesized in the cytoplasm to mitochondria. Remember, there are only about 13 proteins are synthesized in the mitochondria. About 99% of the proteins that are required in mitochondria are synthesized in cytoplasm, and then they are directed to mitochondria. Either to mitochondrial outer membrane or to inner membrane space, or in the inner membrane or inside the matrix. Now what is observed is it has a signal. So they did a very crucial experiment. What was done? So what they did was they took a mitochondria based protein. So therefore this is the mitochondria based protein, and assume that this protein has the signal which is attached at the N terminus. This is a C terminus. Now this protein, it is supposed to get into mitochondria. What they did in experiment number 1, so experiment number 1, what they did was they treat this protein with trypsin. Trypsin, we know that it has got a very specific site for cleaving. So it is treated with the trypsin. So as expected, this will break into smaller fragments. The signal also breaks into smaller fragments. So this was observed in experiment number 1. Then they did another experiment. In the next experiment, what they did was they added energized mitochondria. Energized mitochondria means it is the respiring mitochondria. They isolated from yeast, and so therefore mitochondria is added. So we'll write respiring mitochondria is added, and in the test tube here is the mitochondria, and as per our assumption, our hypothesis, if signals are needed to direct a protein into mitochondria, we will see that if the proteins gets incorporated into mitochondria, and there will not be any protein present in the medium. That is our hypothesis. So therefore, similar to this, now treat it with the trypsin. So if protein is present, if not gone inside the mitochondrial matrix or mitochondria, then trypsin will cleave that protein. But what did they observe? What they observed was, here is a protein, only the trypsin molecules, whatever trypsin was added, only that they could find in the medium. That is a clear indication that if the signal is needed for taking the protein or directing the protein into the mitochondria. Beautiful, a very simple experiment to prove a very important hypothesis. Now comes in the next question. That question is, what is the nature of the signal? Now, in order to prove the nature of the signal, they did a number of experiments in order to prove the nature of the signal. What is observed about the signal is, this signal is about 20 to 50 amino acid length. And in the kind of, so therefore it is 20 to 50 amino acid long, that is the length of the at the end terminus. What is observed is, it has a lot of positively charged amino acids like arginine, okay, lysine. These amino acids are present. Also certain amino acids like serine, they are hydroxyl amino acids, serine, threonine, these amino acids are also present. Apart from this, it has a number of hydrophobic amino acids. So therefore, in the combination of these positively charged as well as the hydrophobic amino acids, it will give, when you make the alpha helical wheel, it will give a positive charge on one side and a hydrophobic charge at the other end. We have explained in a video about how to make an alpha helical wheel. So therefore, on the one side it is a positive charge and the other side it is hydrophobic in nature, and we say that this is an amphipathic. This amphipathic nature is very, very important for a protein to be translocated into mitochondria. This they have experimentally proved by replacing many of these amino acids, and they experimentally proved that this is very important. Let us look at how the protein is translocated to the mitochondria. So what will happen is, if you look at the outer mitochondrial membrane, in the outer mitochondrial membrane there is a protein, and these proteins associated with transporting of proteins to mitochondria are called translocases, and this is a translocase which is present on the outer mitochondrial membrane. Therefore we use the term TOM, and these are complexes. So therefore there are TOM complexes. If the TOM complex which is present here is, one of them is called a TOM 20. TOM 20 is one of the complex. So let us arrange the TOM complex 20 here. So this is the TOM 20. TOM 20. Now the signal from the protein, so this is the protein and this is our signal, and this signal comes to TOM 20. Remember, it is an alpha helical wheel. So it comes here. Here is positively charged and this is the hydrophobic region, and that interacts with the TOM 20 complex. Now this interaction is very weak. It cannot stabilize, it cannot push the protein into the mitochondria. Now this interaction must be stabilized. How to stabilize? There is another protein present in the outer membrane, and that protein is what we call the TOM 22. So here is another protein. Okay, this is TOM 22. Now TOM 22 has negative charges. Because of this negative charge, this interaction is more stable, and it will ensure that the protein is now ready to be translocated into mitochondria. So that is exactly the role of the Tom complex. Now in the inner mitochondrial membrane, it has another complex. So therefore we call that a TIM complex, translocase of the inner mitochondrial membrane. So we draw that. So therefore this is going to be, the TIM complex is going to be here, and it will be almost a physical contact is established between the TIM complex and the Tom complex. And this physical contact is very essential for the protein to be translocated. Which are the components of the Tom complex? The TIM complex. The TIM complex has complex number 23 is very important, 50, 21. These are certain important components of the TIM complex. Certain important things that we need to understand about the TIM complex is in the TIM complex number 50. This is a regulatory complex. Okay, this helps in the TIM 23 to recognize the incoming protein. So therefore, this plays the role of regulation. Whereas TIM 21 and 17, which I have not mentioned here, 21 and TIM 17, they form the channel. So TIM 23 and 17 forms the channel in the inner mitochondrial membrane. I forgot to mention that the Tom complex that has a channel protein that is the Tom 40. Tom 40 becomes the channel. So therefore, Tom 20 and 22, the function is to recognize the protein, bind to the protein, and then Tom 40 will act as a channel, push the protein through channel 40 and allow the protein to come to the TIM complex, and in the TIM complex 23 and 17 becomes the channel, and the 50 helps in recognizing the protein, and it tries to bring or pull the protein or transfer the protein to the mitochondria. These are the steps that are being carried out in order to bring a protein into the mitochondria. So interesting story as you see how you can visualize how the protein will be identified and then it is translocated into the mitochondria. Now we are going to look at what are the experimental proof in order to establish how did they prove all this. We are going to look at that part of the protein translocation.